Okay, so in prokaryotes, bacteria, prokaryotes, they generally reproduce by binary fission, which is a type of asexual reproduction. This doesn't really lead to much variation in the offspring, whereas in us, eukaryotes, we have meiosis and fertilization with crossing over, independent assortment, and so that gives us some variation in our genetics. However, bacteria, because it's asexual, don't have that opportunity. So one of the first ways we're going to look at in bacteria that introduce variation or variety in their genetic information is called transformation. Now, transformation is when some naked DNA or DNA that is just right outside of the bacteria. So like here's some DNA fragments, maybe from a dead bacteria or something. And what happens is the bacteria can take up that DNA. So it actually can bring it inside, as we see here. And then there's two options. That bacteria can incorporate that DNA, which we can kind of see right here, can either incorporate that DNA, and that's transformation. Now that bacteria has a new trait maybe it didn't have before. Or there can be an unsuccessful transformation where the that foreign naked DNA does not become integrated into the bacteria's chromosome. Or we have here, it could also be a plasmid. Bacteria have DNA plasmids, which are just small circle pieces of DNA that are outside of their normal bacterial chromosome. And so sometimes the bacteria will just take in a plasmid like this, and that can also be considered transformation. Both of these options will lead to variety in a bacteria. Here's another picture that we can look at here, where maybe a dead bacteria releases some DNA fragments, and then one of those fragments is taken in to a living bacteria and now incorporated as part of the genome, and now that bacteria might have new traits. An example might be Griffith's transformation lab when he was working with harmless and pathogenic pneumonia bacteria. Okay, so this is transformation.